I'm
Yep.
ready.
Hello. Welcome to Microbiology Journal Club,
Hi.
where we talk about some all things great that are small. Where this week we're going to be talking about a paper about
Yeah.
about how
Um,
COVID-19 manipulates host cells to become into virus making factories, I think, and also how
yeah, a massive paper.
yeah
7 figures and 7 supplemental figures. Most of them go A to F.
yeah this is probably one of the biggest papers we've had to deal with because it covers quite a lot of ground. So basically,
Yeah. Well, I mean, they talk about 3 sections of the host cell, right? Like these are big processes that happen: splicing, translation, protein trafficking. So yeah, I guess it was maybe to be expected.
yeah. So yeah, this is almost like three papers in one, because yeah,
Yeah. Yeah. And it's also something that maybe comes from like the omics, everyone's talking about the big data
mm,
sets technology. Like this is a good example of how like you start with like this really big data set, but then to follow up on some of the things that you see in that data set, like that's what differentiates sort of a good science, right? From like just overview or something like that.
yeah, this really does drill down into what's going on, and yeah. So I think we should just so I mean been focusing on like spike protein and like the kind of bit where it goes inside the cell. We have talked a little bit about this with the replication organelle, which we talked about I think 3 weeks ago maybe. Um,
Yep.
and so this one is another big one. So looking,
Yeah, there's like tons of different proteins that are made, right? In the cell. I guess like maybe the first thing to say is this term open reading
mm,
frame, right? ORF, like that means it's a section of the genome, right? Where they can see like this is where translation would start and this is where translation would end,
yes,
right? And translation, that process of like making a protein. And so you can look at from a genome and you can guess like just by looking at the sequence, oh, there's probably going to be like this many things made. And with the coronavirus, the novel coronavirus, but also other coronaviruses, based on that knowledge, right, we have this idea that there's like 10 to 14 open reading frames, and that might correspond to 24 to 27 proteins.
yeah.
Um,
I mean,
and there's a paper that I've put in the list about like things that I wanted to talk about, right? The coding capacity of
mm,
SARS-CoV-2, where they actually go in and they like try to sequence all of the proteins that come out of an infected cell. And so like this is sort of information you can get at different ways. You can like try to directly observe it, you can indirectly through the genome.
yeah. So I've put up a diagram of the SARS coronavirus genome, um, which
Hmm.
kind of shows you the big like open. So a lot of the big open reading frames, and also in some of the like important proteins. So Spike is labeled
Yeah.
an S. We're all big fans of Spike. Then there's a couple of
We've been talking about Spike a
yeah,
lot. Yeah.
and then there's all these other like accessory proteins. So I mean,
Right. I mean, I think we've also talked about stuff in ORF1, like PL
yeah,
Pro, right? Those are parts of, oh, RDRP,
yeah,
right? RDRP, that's the RNA-dependent RNA
yep,
polymerase. That's the thing that's inhibited by remdesivir. Um, yeah, I guess we've talked about spike. Oh, I think we've talked about E. E is
yep,
envelope. It's like very abundant, and that was like one of the ways in which they measure genomes, I think,
yeah.
right? Like they use uh, RT-PCR against E to say like this is how many genomes are out there because E is like made last, I guess, or something like that. So like it generally corresponds to actual numbers of viruses.
Yeah, so this paper basically looks at the proteins that we don't necessarily think about as much. So there's
Yeah, that's only like 3 proteins we said right now, right? Like there are
yeah, there are
27.
27, and so some of them are well characterized, and some of them aren't. So I think we're going to be focused. So there's the all three and all eight, and they're the less characterized ones. So we don't really know what they're doing. And this paper is going to dig in a little bit onto those. It's also going to dig into some unknown functions of proteins we already know. So things that we think we know their functions somewhat, but realizing that they're doing something else on the side, like they're moonlighting uh, as things that mess with your cells in other ways. So there's this term called virulence
Yeah.
factors, which is a very generic term where it's a protein that does something to mess with your immune system with the host.
Bad
Yeah,
proteins. Well, bad for the host, good for the
virus.
parent,
Yeah,
the virus.
yeah. So
Yeah. And so I guess yeah, that's probably all we have to say about this. Like there's a lot to say about genomes, like viral genomes. Like how do you fit 27 proteins in just 14? Like there's lots of stuff that's just fascinating. But for our intents and purposes, they end up making the proteins that they want.
Yep. So yeah, so in this case, I think for this paper to study the proteins, they took out like a whole bunch of different proteins and then put them into and expressed them separately on these like tiny like I think they put it into human cells or something like that on these.
Yeah, I think they make them in Heck 293
Yep.
cells. That seems to be the common type of cell that they use here.
y-
Yeah, they're just expressing. So like this is synthetic or this is genetic engineering, right? You just like clip out the things that you want and then you make them in another scenario.
Yeah.
And so they make all open reading frames
Yeah.
that are all proteins that they know from this virus, and that's what we're seeing in figure S1A is just the gels. So like they put the DNA that contains the coding sequence of the protein that they want in these cells. They make the cells make them. They blend up the cells and then they run it on a gel here. And they're using like a tag called a halo
Yes.
tag. I'd never heard of this
Yeah, it's just
before.
it's interesting because it's like it basically causes it to be covalently bonded to whatever you want to tag it to, so you can filter it out of a so you can take a protein and filter
Yeah.
out of a specific membrane. I think.
Yeah, like covalent is a very tight
Yeah,
bond, right?
very
Like that's
tight.
like yeah, not ionic, not I don't know what other
Hydrogen
bonds are
bonds
out
there.
there, but
So
hydrogen
yeah,
bonding,
so
right?
of the scale, so covalent bond is like diamond, like ion bonds is more like kind of salt sugar. Hydrogen bonds
Yep.
is like jello, I guess, or like that's kind of how. Don't at me,
Yeah, but
chemists.
these are the bonds that take the most energy to break and
Yeah,
make, right? I think that's the most important. That's the important thing. And so it means that I guess you can purify them really easily. They make better purifications.
yeah,
This is just new tech, right? The tech's always changing.
yeah,
Back in the day, people use like favorite affinity purification of
yeah.
choice.
I mean, the two things like either they get better or less dangerous to use. The thing,
Ah,
yeah.
yes,
So,
that's another good
but I
point.
think this
Yeah.
one is like just it's more easier to use. So
Yeah, I think it's tighter. So yeah, so S1A is just the
yeah,
proteins. So like the darkest band in every lane is presumably the protein, and if you they know the sizes of them because again, it's predicted from the sequence,
yeah,
right? So they kind of say, oh, the darkest band is in the size that they see. I don't know the size of all these proteins,
yeah.
so I'm not personally verifying this, but I'm gonna trust them on that one.
Yeah, I mean, I'd be scared if it was like too clean. There's always some mess in these protein bands, so
Right,
yeah,
right.
it's almost reassuring to see that because proteins again they have different conformations, they can run different levels, they can fragment, there can be other proteins that look kind of similar. It can be a little bit
Yeah,
messy, but the important thing is like they've got something in there.
right, right.
So we can move on to the
Yeah,
next thing, which describes about the so figure 1A, which kind of describes their method for figuring out what each proteins are binding to. So they know that there's
mhm.
like SARS-CoV proteins out there, but what are they binding to in the host? Are they binding to host mRNAs? And or like yeah, I think that's what they're looking at.
Yeah, I think that that's what they want to know. Like they want to know this is the question that opens up the
Mm,
paper, right? Like what and it's because they have the capability to do
yeah,
this, right? Like it's also born from that. It's like this lab knows how to find out what proteins bind to mRNA and where,
yeah,
approximately, right? They can get an approximate where. And so they do that, well, you can see in this diagram, right? Like they do that by cross-linking the proteins with UV to the
yep.
RNA.
So,
And then they digest it partially and they put it through the favorite technology of this era in science, right? High throughput sequencing.
yep. So the thing about UV cross-listing, because it's something that I hadn't really worked with very much, but it's quite interesting because because
Mhm.
each like of these so the protein and the like RNA they have like ring molecules that absorb energy from UV and they
Mhm.
can be induced to again covalently bond to each other if you add UV light at like fairly low energy levels.
Yeah.
And theoretically, you can do this with lots of different proteins, lots of different levels. So DNA does it but at much higher doses. So that's why you don't want to be in the sun for
Mhm.
too long. But this one they do it at a low
Yep.
dose, so they can actually pick out. So the protein just gets permanently bound to that point in the RNA and cross links.
Yep. Yeah, so that means that the section that the and maybe they oh yeah, that means that the section that the protein binds to is going to be occluded, right? The RNA there is going to be like
Yeah,
gummed up basically
yeah,
because it went through like a chemical reaction to fuse with the protein. And the spaces that aren't directly touching, those are up for grabs, and you can sequence those things. So you can imagine, I guess, like, yeah, the diagram sort of shows us that, right? Like, they have the example of RT1 and RT2, right? Like, those are examples of fragments that can get read, right? And as those fragments accumulate, right, you'll see that they stop at certain
yeah,
points, right? The fragments always end at certain areas, and like the magic of computers then like builds us a nice graph that shows us the frequency, right? That a fragment would stop at that
yeah,
point, and you get the sense of like where the proteins are binding from those spikes in the frequency of stopping.
yeah. And I mean, it's really interesting kind of technology, and it's kind of fun because you're almost
Yeah,
like like you're counting the losers, the people couldn't make it at the end. Like, where are they tripping over before they get to the finish line? And
right,
they're tripping over the point where they like cross-linked. So there you go. Okay, look at
right.
all these loser RNAs that didn't make it. There's like an obstacle there. That's where they're tripping over.
Hmm. Right, right, exactly. And that's the same. This other figure is like just from another paper. It's the same technique, right? The technique called enhanced cross-linking and immunoprecipitation, or
Yes.
ECLIP.
So this is another
uh,
technology that kind of does a similar sort of thing, except they digest like the RNA, then they digest the
it's
protein.
it's the same. No, this is the same. Like I just picked
Okay.
this out from another paper because I thought it had like good details, right? To like because not everyone would have seen from the previous figure like some of these things. Yeah, you have to digest it still,
Yeah.
right? You have to break up the protein or the RNA in different ways. You can see they split them into two
Hmm.
groups, right? They have a group that they did cross-link and a group that they didn't
Right.
cross-link into. Yeah.
Yeah. Okay.
um,
And I think that
so like that's part of the algorithm, right? The algorithm looks at the input versus the output, right?
yes.
And through understanding the difference in where they see those fragments is where they build that graph that has a little stop points and stuff.
Yeah. So
um yeah, and then the other thing that this I like that this figure showed that the other one didn't is that it shows like this whole library prep thing, right? So it's like it's one thing to have the fragments, but then actually there's tons of processing that's done to make like a molecule set that can go into the machine that reads it. Yeah, so it's just like it's a complicated process, but we kind of get to see the fruits of that, all of
yeah.
that.
And so the next figure is almost like the controls to see set up.
Yeah. Yeah, so they're so PTBP1, that's a known
Yeah.
protein that binds our
Yeah. I believe
mRNA.
that's called
I
pyrimidine
think it's a host.
processing.
Yeah, it's a host
Yeah.
protein. Um, and this is because yeah, there's so many mysteries in cells, but like there are these. It's not a mystery, but it's known that like inside the host, mRNAs can get aggregated to control their expression,
Yeah.
right? Because if you group all the mRNAs together, then they can't be accessed by ribosomes. So there are proteins that specifically bind mRNA in the cells. I think PTB1 is one
Yeah,
of them.
pyrimidine,
Uh,
pyrimidine tract binding protein is what it stands for those in the audience who are interested. Um,
Oh, I mean that's nice because in D, right, it's like that's what it binds to,
yeah,
right? Like they're showing that they can see from, I guess, the cross-linked area, right? Like what they think that the binding site would be for this PTB1B, and I guess that correlates probably to the actual one. These are all controls,
yeah,
right? Where they're saying like their stuff is working. They can get at an approximate binding site for these
yeah. This is
things.
like the normal world if we're on the hero's journey version of storytelling, where before we go into
Oh
the
yeah. Yeah, before we dive into the unknown, like let's just make sure what we're doing gives us what we do
yeah,
know.
yeah,
Yeah. And so I think they also, yeah, they can figure the halo enrichment, I think it matches, right? Like the things that they see that are drawn down um by their sequencing uh strategy, it's the same thing that gets drawn by down by the proteins
yeah.
itself, right? When you grab the proteins from the
So
affinity tag, the
yeah, you
halo.
get more signal to better signal compared to noise, which is a good thing to have because this
Hmm.
is you're dealing with very
Hmm.
subtle small things. So being able to isolate your signal is very important. And
Hmm.
yeah, is there?
Yeah, I think that's
Yeah.
good.
Um,
That's all I want to say about that.
Okay, cool. And now this is probably the most important figure of the paper because this sets everything up for what we're going to go doing.
It's Yeah, it's what they found out.
Yeah,
This is the biggest. This is from that screen of all 27 things. They listed them there on in B. They all go down. There's the NSPs, there's EMN, and then there's those unknown ORFs. Um, and they made each of those successfully, as we saw. And then they did this procedure where they tried to find the mRNA binding sites for
yeah,
them, right? By looking at the fragment, by looking at the difference in the frequency of fragments bound and unbound, and that stop site.
yeah. Pew, 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 seeing what sticks and what doesn't stick. And they
Yeah,
found that,
and from that a heat map where black is the most enrichment and white is nothing.
yeah. And they do it on a whole raft of different host mRNAs. But the one that they noticed there are a few like really important ones that they decided to focus on the paper.
Well, yeah, they don't have to focus, right? They don't have to focus actually. This is the so-called unbiased
Yeah,
screen, right? Because this is all the mRNAs that exist in a cell that they
yeah.
use, and I think they're using like again heck 1, 2, 3. They'll switch cell types a
Yeah.
lot, but their original screen is in one
So it'll
cell
go
type.
between like human
Uh,
cancer to human embryonic kidney cells to vero cells. I think there's a three
yeah,
main cell types. So it's one of those.
yeah.
Um,
Yeah, that they used here. And again, the sequence of those cells are all known, so that's why they can pick out whatever mRNA is touched because it's all done through sequence,
yeah,
right? The physical element was the cross-linking between the protein and the mRNA, and that's going to get outputted into our database of everything that we know about mRNA, and then yeah, give us the associations.
yeah,
You can see in C, that's the example, right? Like, so because it's a big, you can just go through everything, right? Like in their database, right? For all the sequences that match up, or for everything that was treated with NSP9, right? Like here are all the sequences that match up with COP S5 or HOX C10 or OBI1.
yeah. And it just
Actually,
uh,
the next figure would be the same,
yeah. So
right? Like,
effectively, like this is like the line indicates like the area of the gene. So like from front to the end, and then the spikes indicate
mhm.
like how much the mRNAs are being like bound at that point by the
Yeah.
NSP13 or NSP12. I mean, yeah,
Yep. Each of them, they have it for us. So like you can imagine that this B actually represents like a huge amount of like C graphs,
yes,
right? At every point here, at every point where the protein intersects along, right? The protein comes into like some color, right, along the graph of all these mRNAs. That is one of these graphs that you can see in C. Yeah, it's all condensed,
yeah,
and the color is made so that we can just like from a outlook look at these things.
yeah.
And so I guess they start discussing in the paper like the patterns that they're seeing, right?
Right. Yeah, about like where the peaks are happening and why they're happening there. So I might have got figure
Yeah.
S1F up where you're looking at some of the proteins that they decided not to look more deeply in. I think because and
Mm-hmm.
but then if we go to
I only recognize JUN. I think JUN is some sort of immune
yeah
regulation.
yeah I
Yeah.
reckon that's the only one. I mean I think I reckon yeah I
It's like part of it's part of some sort of kinase
yeah
thing. That's like,
there's a bunch of there's a
like
really important signaling cascade of different proteins that involve so many different proteins. I can recognize the name of it, but I cannot remember where in that cascade it comes to. So
yeah, or what specifically it controls, but it's like something like that. That's like some other
yeah,
signaling stuff. But in the paper they say some other things. They say they find things involved in protein translation, protein transport, protein folding, transcriptional regulation, and immune response. So they find a whole bunch of
yeah,
things, right? Those are all domains, and that's all knowledge based on the database of what we know, right? Like this is built on the backs of so much other science that's come out, right? Saying like, oh, we found this is involved in this, and this is involved in this. And the cool thing about this approach is, you know, just by finding the physical, right, the physical association between these SARS proteins and these mRNAs, now they can start inferring things like, oh, does SARS-CoV-2 affect um, immune immune response, protein transport, protein translation, protein folding?
yeah. So they do start to focus in. So going back to B, they start to focus in on U1 and U2, which are very important
Mhm.
proteins for this thing. I believe it's called the spliceosome. Am I?
Spliceosome,
Yeah.
yeah. Which is basically like the combination of all the proteins that come together to do splicing.
Yes.
And for those who don't know, intron for us, eukaryotic human beings, introns and exons, they're regions of our genes, and they aren't just made, they can't just be read directly from our
Hmm.
DNA. They actually you have to remove some extra sites.
Yeah.
Um,
So
yeah, the
so
introns.
genes usually encode proteins, but there's a lot long spans of the genome that aren't meant to encode proteins that are there for other structural functions. Sometimes they're there so you can have alternate versions of proteins. So, but in this case, you don't want them to be split. If you want to get a functional protein, you want to get rid of these introns because they have other functions that aren't necessarily useful for the rest of the gene. So
Yeah. Actually, I think that's a mystery, right? Like I think that those are ongoing theories of like what are they
yeah,
for?
I think the because there are like I think there are multiple reasons. Because some introns happen because a virus integrated in the genome and then lost some function and then ended up just either disappearing or becoming a functional
Yes.
part of the genome
Yes.
sometimes, or sometimes it's like a gene gets copied during transition, or I mean sometimes like there are things.
Yeah, there are many reasons for them to arise, but then in every instance of a gene, evolution's acted
Yeah,
on it, right, over time.
so
Right? The introns have appeared over time and then evolution acts on them. So like they fulfill some purpose being there. Like I think I've also heard that they prevent like damage, like if damage comes down, like less of the region of the DNA is like important
yeah,
or whatever. Um, yeah.
so
So anyways,
they're really interesting.
before we make proteins, we generally have to excise those introns because they aren't used in the coding
Yeah,
sequence. And there's a complicated machinery inside of the nucleus that does
yeah,
this,
and that is called the spliceosome, where
the
they snip up
spliceosome.
the genes, they snip out that RNA before it goes into the ribosomes.
Yeah. Yeah, so these two sets of figures, 2AB and S2AB, this is looking at NSP16 first,
Yes.
binding to one of the spliceosome um, uh,
Yep.
mRNAs. So, like, I think maybe that's another important thing that we don't always talk about, or people may not be aware, but mRNAs are also part of, or not mRNAs,
Hmm.
RNA, right? Because mRNA implies
Yeah.
messenger, that it like makes a protein. But RNA itself is part of some of these molecular
Yeah.
machines. And like we'll talk about it later, but like the big molecular machine, the ribosome, the thing that makes proteins, is actually a grouping of protein and
Yeah.
RNA. And similarly in the spliceosome, there's like proteins and RNA. So the method that they use gets at RNA. So we're just seeing the RNA
Hmm.
part of all of this, right? Like we're seeing, oh, this viral protein touches these RNAs that are important in these big machines that exist in our
Yeah.
cells.
And as you can see, there's this big spread spike in the early part of the graph. So they're binding to a very specific point on the U1 RNA. So the question is, what is that doing?
Yeah, very
Yeah.
specific. I think they model it out in B. They show it's actually this very small site that they presume that it binds to based on that
Yes.
peak.
Uh,
And in the next one, this is NSP16 again, but now with the U2, so another splicing
yeah.
mRNA.
Uh,
A little bit noisier,
Of course.
this
Yeah,
graph.
we'd expect that from you too. I mean, the Joshua Tree, very noisy.
And again, you're seeing the same thing. Three nucleotides is what they presume is this binding site between
Yeah,
NSP16.
so these this one protein targets two different mRNAs, and it does seem to be fairly specific for an
RNAs, just
RNA. Sorry,
RNAs,
RNAs.
right?
Sorry, yeah,
Yeah.
these U2
Mhm.
RNAs. I think they're like yeah,
Yeah.
I can't remember the exact correct term for them, but yeah. So this is kind of an important thing for this.
Yeah. Yeah. I guess, yeah, we should say that because in these two figures, they show the mRNA, right? That is this figure in B, they didn't collect this data, right? This is just like them showing us details from stuff that's already known about U2 and
Yeah.
U1, right? U2 and U1, they have to do splicing, so they work on
Mm.
mRNA to remove the pieces that you don't need and stitch them back together into the full message. Yeah, I guess that's important to say.
Yeah.
So we know, so from this is all from that first experimental approach, right, where they looked at the whole
Yeah.
set of things that bind to RNA, and then they zoomed in and they said, this is really interesting. Here are two spots that NSP16 binds to. They're the same spots that bind to mRNA during the splicing process. So there's a hypothesis that you can put out there and say this protein inhibits splicing.
Yeah. And I think the next set they're going to try and figure out like they're trying to look at that, characterize it. So
Yeah, they're going to confirm a little bit
so
better.
yeah, this time they're using real SARS-CoV-2 in vero cells. So those are from green monkeys,
Mm-hmm.
if people are aware, and seeing like using an antibody against uh, the NSP16 to see where it's localizing in those cells.
Yeah, yeah. And they make the antibodies for this paper,
Yeah,
right? So I think like we talked about last week, like, making antibodies for therapeutics. In the research community, you don't have to worry about some of those stringent like safety things and like
yeah,
efficacy, right? We make antibodies all the time because like it's like a major tool in order to see
yeah,
things. And we've seen pictures like this before, right? It's always antibodies. And like when you're working with a new protein, like proteins that have never been worked on before from SARS, you got to make the antibodies
yeah,
yourself. Yeah, so they inject them into rabbits, and the rabbits make the antibodies, and then they take the rabbit blood.
yeah,
Um,
and it, and they get
so in these, yeah. So what are we seeing in these? I guess we're seeing where is the NSP16 in the cell, right? Because they have cells. They're looking at where it is, um,
and it seems to be
and
localizing to the nucleus. So inside the nucleus, where
yep,
a lot of the splicing is happening, because this is happening in the process of the mRNA being created before it goes outside to the rest
yep,
of the cell. So if it's affecting the
yep,
spliceomes, this is where the NSP needs to be in order to because if it
right.
could easily bind these things, but if it doesn't actually go to that place, then it might not be functional at all. So
Absolutely. Yeah, you got to confirm. And this is sort of the thrust of science in
yeah,
general, right? Is like getting closer. Like, yeah, we saw something that suggested this, but how do you really know that it does that
yeah,
thing, right? Let's go through like lots of these
because yeah,
possibilities.
there's always coincidences do happen in science. So trying to isolate that as
Hmm.
much as possible because you don't want to
Yeah.
be like proven.
Coincidence
Yeah,
happens all the time.
yeah,
Science and otherwise. There are so many coincidences in the world. They're not all meaningful.
yeah, and that's why we have
uh,
statistics and all these other things to try and like make sure that we don't go down a rabbit hole that isn't there's nothing there or so and
right.
while we're doing these
And in this case, they're using well, we also use controls, right? They have NSP1. This is something that they don't think would go to the nucleus, right? And so like that's used as the contrasting
yeah,
here, right? That say like, oh, and here we stain for another protein that we think doesn't go to the nucleus, and sure enough, it
yeah,
doesn't.
and we'll be talking a bit about NSP1 later, but but it's quite important to because I mean you remember like a couple of weeks ago when he did the spliceosome? I feel like I'm seeing not spliceosome, the replication organelle. Like I feel like I'm
Oh
kind of
yeah, the
seeing a few of those
replication.
patterns with the NSP1, but I could be seeing things. So, um,
Mm.
but yeah, no, I really like this one thing.
Oh, like you think that the NSP1 might be staining as if it was in those membrane
Yeah,
bound
that's what I was
things.
thinking, but those things tend to be more
Yeah.
connected to the ER rather than the endoplasmic
Right,
reticulum rather than the nucleus directly. So I might just be seeing
right.
things. Again, one of those things where you can go down a rabbit hole with these sorts of things and start seeing things that aren't there.
Yeah.
So, but it,
But so that would be right. That's the domain of like, if you had if you had your own lab, right, and you were reading this paper,
yeah,
right, and you had these capabilities, you might see this and be like, oh, we should try this, right? Like I'd like to see if it correlates, right? If NSP1
yeah,
correlates with this other protein that I know
yeah,
about.
you'd write it up in a
Um,
grant or ask your PhD student who has grant money to do it for you and see whether
yeah, yeah. Okay, yeah. So, anyways, takeaway from this is that we're figuring out where NSP16 localizes. Seems like it's in the nucleus in infected cells.
yeah. And figure S2G also looks NSP16 as well. Uh,
Yep. And but this time they're not infecting them; they are just expressing them in the HEC293 T
again,
cells. So yeah, Vero cells are from monkeys, right? And HEC cells are from us,
yeah, human
humans,
embryonic
embryonic
kidney
kidney
cells.
cells.
So,
Yeah.
yeah, this is important because again, these proteins that we're looking at aren't exactly the same as wild proteins. They've got this halo-like protein that's attached to them that might make them behave
Hmm.
differently. So trying to
Hmm.
confirm that these two things are behaving in a similar way is quite important because yeah,
Absolutely.
um,
Yeah, they talk about Delta RC later, so we won't talk about it
yeah,
now.
delta RC. That's something to note down a little bit later, but
Yeah. So we'll move on.
yeah,
So yeah, so they know that it does from their sequencing, they know that it binds, and then from these pictures, they know that it's in the nucleus. So here's their little model
yeah,
that they think is happening, right?
the
Normally, mRNA is going to be bound by these two U1 and U2s, but if NSP16 is around, it binds those sections instead, and you don't get splicing.
yeah, bingo. So that's the NSP16 paper, I think, nearly done. So oh no, no,
Oh no, no, no,
they're
they're
doing
gonna
more.
keep going. They're gonna keep
Good.
going. Yeah. So they make a
Hmm.
test now, right? An explicit test, right? Because like all of this is just being correlating with location and with the binding event. Let's like test that theory out, right? They have a reporter. So this is artificially created, right? An artificial gene, right? That has the exons, right? Are split up by this intron that has a stop on it. So normally when this piece of mRNA goes in a cell, the ribosome is going to bind to the beginning of it and read along, and then it's going to hit the stop, and it's
Hmm.
done,
Yeah.
right? But in the nucleus, there should be splicing, and it should remove the intron, right? And by removing the intron, then you would be able to continue down that line and make GFP, which is what happens at the end. So they put these reporters in, and they see if NSP will stop this event from happening, right? So if they see less fluorescence, they know that there is something inhibiting the splicing
Yeah. So
because our cells don't normally glow green.
yeah.
If that's not clear.
So yeah, so basically in figure 3b and S3a, they show this they have a control where they have cells with gap DH and cells with NSP16, I think.
Yep.
And
Yep.
look,
Yeah. So again, they're making NSP16 in the cells. This is just like G, right, that we were looking at, and they cell sort them with the reporter.
yeah, and they find like
And GAPDH is just like a regular old protein that exists in our
yeah,
bodies, an enzyme.
yeah, it's something to do with the cytoskeleton, I think, or I can't remember exactly what, but it's just everywhere
Yep.
equally. And so they find like that.
Yeah, and they get less.
Yeah, so the peak basically like shows that there's a lot of low intent GFP intensive cells in the NSP16 cells, whereas in GAP DH it tends to be like a more broader swell of like cells.
Hmm.
So they try to like
Hmm.
sort it out between like seeing like exactly how. So lots of NSP16 with like a level 3. So I mean, thing is, cells do sort of slightly fluoresce green. So
Yeah.
there is a fair bit of
Yeah.
background. So that would be probably a lot of the signal would be from that. But then,
Yeah.
so
Well, I think the other thing is that there's just a variation. Like the reason why the gap DH is so broad is that that's what you're trying to
yeah,
explain, right? Like why why so broad of a fluorescence spectrum?
that
I mean, I also think that it's probably because they don't have non-reporter cells in this,
yeah.
right? So non-reporter cells may be even lower
Well,
than 3, I would
I
say.
mean, like, yeah. I guess like the non-reported cells would basically show a normal distribution about the same peak as where NSP16 is peaking. That's what I'd kind of say. And because there's an extra
That what you think?
fluorescence,
Yeah.
that's pulling the gap DH a bit further out.
Yep. For sure. For sure. For sure.
Um,
Uh, yeah.
yeah. So
So that's frequencies of cells, right? So like every cell could be slightly different because like things don't happen the same in
yes,
every cell.
and it's actually really important to recognize this because sometimes when you look at bar charts, you kind of can forget that you're looking at all these different things, and that you can,
Yeah, they're averaged together into the
yeah,
bar, but really like it represents like a heterogeneity. Yeah, I mean like this is I don't know if people are familiar with the whole single cell technologies that people are doing. Like now people like they don't want to average things anymore. They want to know from each cell, like just give us the data from every single
and
cell. Anyway.
that's kind of I think that's a good thing because I feel like just by going by averages, you can lose quite a lot of signal, a lot of interesting things that are
Yeah,
happening.
absolutely.
So it's a good thing.
Absolutely.
The only hard thing is like interpreting it because all of our statistics are based on averaging and those sorts of things. So trying to interpret these results to figure out what model works and what doesn't, that's going to become a bit more challenging. Which is, but that's
Certainly.
science. Oh god, it's going to be challenging. Oh no, but that's kind of what science is. So
Okay, let's see. Well, let's keep going here. So now they're going to do another
Yes.
test, right? They're going to look at, they're actually going to directly measure the proportion of unspliced
Yeah.
fragments that they see. And they do that because they're labeling, or how they're going to see RNA is they're going to label it with this 5EU, some sort of radioactive nucleotide
Yeah. Let me
modifier or something like
see
that.
whether, because I think it's some fluorescent. Because let me double check what 5EU does. Uh,
I'm not sure if it's, I think it might be that it binds biotin so they can grab these
yeah, I think it's something
pieces.
like that. I mean, like,
Yeah, it's just
I
about
mean, as of Britain, as
grabbing.
someone who lives in Britain, I do expect EU to be radioactive, but I mean, it's. But yeah, I think it's something like that where five E.
Well, in B, they're saying biotin 5EU conjugation.
Yeah.
So like that's like
So
okay, they're grabbing the stuff that they want.
yeah, yeah, it looks like that. So that's basically because biotin grabs stuff and it grabs it
Yep.
really tightly. So
Yeah.
um,
So what they're doing is they're labeling all the mRNA that's being made from ribosomes. They're grabbing a bunch of
mm,
it, and they're able to enrich that every time they do it to get more and more of these nascent mRNA fragments. And they're going to sequence it, I think.
yeah,
And they're going to sequence it and they're just going to see, are they spliced? Are they not spliced?
yeah,
Yeah, because again, we know the sequence of the cells that we're working with. So we know like what the introns and the exon pieces are. So by going through this, by just collecting all the RNA that's being made, fresh RNA that's being made, yeah, they'll be able to just measure how much splicing is happening by looking at the
yeah,
sequence. Okay, so they do that in these violin
yeah.
plots.
These. So yeah, you've again going pretty much all single cells. I think looking at all the data and seeing that like again the percentage of unspliced.
Per gene. That's
Oh, per
single
gene. Oh
cell.
right.
That's per
Oh
gene,
right.
right? So yeah, all the dots are for every gene in the cell, or every piece of mRNA, right, in the cell. We'll
Yeah.
say that's probably the best way to think about it. I guess they say per gene, so it might mean it's binned. Like maybe there's many RNAs that get made from a
Yeah.
gene, so they're gonna just collapse that into a single data point. And again, they're looking for is in everything that they sequenced, did it have introns or did it not have introns? And then assign that frequency number into every gene in the cell.
Yeah, and I guess the thing that we're looking for is like there being like a fat top to like almost looking like a Christmas tree where you because usually like you'd want it because for normally you'd have things circling around the middle, but if things are getting pulled upwards, then we're
Yep,
seeing more unspliced. Uh,
for sure. These are basically frequency graphs like turned on their side,
yeah,
right? And then played out for each condition, right? And they have I guess they have replicates, right? C is like different replicates of
yeah,
them. They've done it many times to show that like they consistently see the
yeah.
effect.
D is like the pooled version of
And
C.
it's
Um,
yeah, and D is the pooled version of C.
oh, sorry,
And so
yeah.
yeah,
And sorry, and in the little corner there's an SD as well, which is looking at NSP16 versus GapDH and the other NSPs to see whether they
yeah,
having effect on the splicing. Apparently not. This seems to
yeah,
be something that NSP16 is doing.
yeah. So again, a very important control, right? Because like controls are all about basically um, directly addressing some sort of like simpler
Mm.
explanation, right? Some sort of non-interesting,
Yeah,
simple explanation, right? So like you could be like, oh, but that's just like viral proteins. Viral proteins just love, like just disrupt splicing when you infect a cell, right? Well, they'll show us. Actually, no, right? It doesn't happen with NSP 8 and 9. Yeah, and I guess like the important thing is like this is messy, right? Like this data, not all splicing is disrupted. Some of it does fall under, right? Like is at that zero mark where like no change,
yeah,
right, in splicing. So like this effect isn't like we don't know. We know the effect happens maybe through this mechanism, but we don't know if it's specific for certain genes or it's global. I guess this would suggest that it's somewhat global because a lot is
yeah.
being shifted upwards, right, into the
I mean,
unspliced
this kind of does take
percentage.
a very global view of things, so they don't. It's not like they
Mhm.
pare down to individual genes. They're looking at everything. So
Right.
Which and it's very
Well, I
impressive.
mean, from this data set, you could pull individual
Yeah.
genes out and you could ask, right? But that's not what they're showing us. They're showing us everything. And they also do it. The next figure, E3E and
Yep.
S3E, they're just doing the same thing, the same assay, but infecting the cells with
Yeah. So
SARS-CoV-2.
does it happen with real SARS-CoV-2? And it does. Apparently, seems to
Yeah.
happen more expressively. So it might be that
Yeah.
the NSP16 isn't the only
There
one.
are other things work together.
Yeah.
Yeah, other things work together to make this effect. Okay, and then oh, I see. They're also just looking at in
Yep.
S3F, they're looking at just overall mRNA
Yeah.
levels, right? How are they affected upon infection? And they're
Yeah.
down. Um, and I think this is supposed to be consistent with the idea that there's less
Yeah.
splicing because RNA that isn't spliced is
Yeah.
defective. And so it would be targeted for degradation by the cells. And so this is like a consequence, right? They sort of said, okay, well, if splicing is affected, then what's a consequence we could see of that? Well, all the mRNA should be down
Yeah.
then, because the cell's like all this
Yeah.
junk that we're making, we gotta get rid of it. And sure enough, they're seeing that same effect here, right? That the mRNA levels are down. So I guess we're into their
Yeah.
model. The end of the first section of this paper, part one, is they think that this NSP16 goes in, binds up these U1 and U2s, which means that the spliceosome can't recognize the introns, can't do the splicing. You get all these random unspliced mRNAs accumulating, and they just get sent to the
Yeah.
bins.
They just get queued up and thrown out because
Yeah.
the COVID is messing with how mRNAs are being created. And so that's
Hmm.
yeah, this is part one. This is A New Hope is over. And now we're going to go to
The Empire Strikes Back. Except it's like it's all the Empire Strikes
yeah.
Back.
Oh wait, actually no, I forgot the next one. They do link it to the actual host defenses.
Oh yeah.
So
Oh
yeah,
yeah. That's kind of nice. Yeah. So the one reason why they think they want to give it
yeah,
relevance, right, to the infection. Yeah, they want to say, "Oh yeah, why does it even matter? Why would it matter that the
yeah.
virus wants
It
to break
downregulates all
down
the
all
proteins.
those?"
Wait a minute, isn't
Yeah.
interferon a protein? Yes, it is. Let's see where that gets downregulated too. So
Absolutely. And we'll see this tool come back, the ISG
yeah,
reporter. That's a cell line that responds to interferon and creates, I think it makes, I'm not sure what it makes. I think it makes fluorescence potentially. But anyways, it makes something in response. And you can measure
yeah,
that. And so interferon response, well-known antiviral pathway in our cells. And they see that if you add NSP16 to these ISG reporters and then you stimulate them with interferon, they don't respond as
yeah,
well. And that's the same sort of thing that you could see when you use a spliceosomal
yeah.
inhibitor in
I mean,
disease. Yeah.
theoretically, you'd do it with lots of different inhibitors, but yeah,
Hmm.
you can see the story they're trying to put together. This isn't necessarily the most like well thought out version of their paper, but it's not really the point. This is kind of so the a doctor might read it and go, "Oh, is this something to do with the immune system? This might be interesting." But it's kind of almost like clickbaiting
Yeah.
people into reading this. But
Yeah, yeah. I mean, this is, it's challenging. Like already we've talked about all these experiments that have been done, and it's only part one,
yeah,
right? So like this is just like a little sliver to try to make that information that they told us maybe more relevant to an actual disease process,
yeah,
right? They're like, okay, so what? It hits this big global thing. Well, they're suggesting here,
yeah,
right? Just lightly showing us that maybe it has something to do with interferon.
yeah.
The link's not well established, but it's there. They
I
planted
mean,
the
yeah,
idea.
I feel like the idea is there. I mean, because all the immune system, all the stuff the cell produces that are anti like viral are also proteins. So if
Yeah.
you get rid
Yeah.
of all the proteins, then
Hmm.
theoretically you make things less inflammatory. Although immune system is so complicated, you probably have a paper that says the opposite, and it'll be like, oh, that
Yeah.
also helps the virus as well. So it. I mean,
Right.
I think we saw
Right.
a super antigen paper, which again causes inflammation. But yeah, it's so
Right. There's some balance, right? It's always complicated, like sort of like yeah. I was gonna say what parries, stabs and parries,
yes,
right? Like between the two sides, and somewhere in that mix, you get like what the disease is.
yeah,
So in 3H and S3H and I, remember we were talking about those violin
Yeah,
plots, right? Those represent like a whole bunch. They just pick out the immune genes from the violin plots, and they show us what the splicing versus unspliced
yeah,
is. And so, like, okay, when you go into their data set with the eye for immune genes, you do see the effect that they're inhibited from
yeah.
splicing.
I mean, if you shut down power to the city, the police lose power as well. Therefore, it works. Kind of,
Yeah,
yeah.
yeah. Hmm.
And now we're going all the way back to the first figure 1 BC. This time we're looking at uh,
So go to point two.
18S binding. So 18S is a part of the ribosome, which is very important
Hmm.
part of the cell. It turns mRNA, reads that code, and turns it into protein.
Hmm. Yeah. Again, it's a huge thing. I think maybe five. Oh, it's been
Yeah,
crystallized. They like have a big structure of the protein, but it's really big. Oh, here, there's a good picture of it
yeah.
in the next figure.
It's
Um,
it's really important to the cell. I mean, it's one of the things that in rough endoplasmic reticulum, they like you take a picture. It's called that because it's like a normal membrane, but it's got the ribosomes hanging off it, so you can actually see these under a microscope, or at least see the effect of them.
yeah, under an electron
An electron
microscope.
microscope, yeah, not a real microscope, unless you squint really hard.
Yeah, yeah, yeah. Yeah, so this is a big machine in our cells make stuff. 18S, they map it onto the big structure, and you can see it's in a very important
Yeah,
location. 18S sits like right where the mRNA it goes into the
yeah.
ribosome.
So
And so again, maybe if people aren't super familiar, we can use some analogies, right? The ribosome is like it reads, right, the message from the
yeah,
RNA. I guess maybe that's
it
not even an
literally
analogy.
it
That's what
has a
happens.
hole. The mRNA goes into the hole, and oh god,
Yeah.
what am I doing? Uh, it goes into the hole and kind of lines up other complementary like amino acids to it, and then links those together
Yep.
in the chain. So almost
Mhm.
like it's like an assembly line where it just like
Yeah, it's a factory. It's reading information that comes in. Yeah, I don't know. I always think of it. I mean, I don't think this is a good
no.
analogy because it's not super relatable, but I think of it as like a Turing machine, right? Like the thing that reads the paper, it reads like just one by one. It can only go forwards or backwards, and then whatever it sees, it does that thing,
So
right? It's like a type of
you're
machine.
welcome, any computer scientists who are watching. You are welcome for that analogy. We get you.
Yeah.
Other if that person watching us can create an analogy for how the Turing machine works to normal people, then we've got this level of translation that people can understand what we're talking about. This is science communication at its finest.
Confusing to confusing. Yeah, don't
Oh
worry
no,
about it. Got you covered.
oh, I mean, in terms of confusing analogies, I've come up with more confusing ones. I intend to. It's like a deliberate part of. So it's like a dolphin that swims into a tunnel, and then but dolphin's got stuff written on it, and the thing in the tunnel reads it, and then it stumbles. I mean, it's you. I mean, I think yeah.
Yeah,
So we're not the first science show to have
it can be
terrible, weird
bad.
analogies. I think it's part of communicating science. Be-
Okay, yeah, let's move on. So, yeah, so it binds this area of 18S, and they know other things that bind this
yes.
area. Um,
So other things like
and
SIRBP1 and like a couple and
yeah,
STAN1, which are also things that are bad for ribosomes working. So it basically blocks.
yeah, they stop
Yeah,
translation.
it blocks that hole, so the mRNA can't really travel through properly. It stops traveling through, and you can't make protein anymore.
Right, right. So again, that sort of drives the hypothesis. Maybe NSP1 blocks
Yeah,
translation. So they do that with HeLa lysates first. So they just like blend up a bunch of HeLa cells,
yeah,
poor HeLa cells, and that mix of cell juice, right, would normally make
yeah,
protein, right? If you put in some GFP sort of expression vectors, it would make that protein. This is very biochemistry. Then you add some NSP into that mix, and you get less of that protein.
yeah. I mean, protein. Where's the protein gone? Oh yeah, add NSP. It's gone. Just it's yeah.
Yep.
Uh,
So they show us in
yep. They
cells.
show.
This is figure 4CD, and they have cells that would normally make GFP, but here are these the HeLa cells?
No,
Oh no, these are
HEC
HEC
293 T
293
cells.
T cells. Yeah, because that's where they make all their proteins. That's the
Yeah.
protein expression.
So yeah,
Um, yeah, and it's not made. And they also do the expression with NSP 8 and 9, right? That was their negative
yep,
control in the last one as well. They also add an M protein here, I guess another negative control just in case.
yep,
It's NSP1 is doing this, and it seems specific for NSP1.
yeah.
Okay, then they use this
Sunsets.
this new thing. This is new to
Yeah,
me.
surface sensitive.
Um,
This is a new one for me because back in the day we'd use radioactivity for this. You'd incorporate
yeah,
radioactivity into
this
like your cells, and then you'd use that to measure like what like.
how much what new proteins are being made.
But instead
Yeah.
they use
Yeah, this is
sorry, you
I was gonna say you alluded to this earlier in what we were talking, right? Like one of the ways in which technology marches on, right, is to make things safer for
yeah
scientists to use. Yeah, and it's nice because also if you when you're using radioactive isotopes, you also have to be close to a source of radioactive
yeah
isotopes, and that's not true everywhere.
yeah.
Yeah, that you'll be able to
You
get
have to
that.
like work very fast because otherwise you lose your radioactivity. Whereas this one, pyromycin, is like a new. Oh, I guess it's like a tRNA analog or something like that, where it's produced by
Yeah.
like
Mhm,
bacteria,
mhm.
and it's been used as like an antibiotic, but also is kind of harmful for
Yep.
humans as well. So yeah, don't
Yep.
eat it, but it's
Don't eat
yeah,
it.
don't eat your experiments. I mean,
Yeah. I mean, it's actually used inside of selection for these
yeah,
cells, right? So like they're making these cell cultures and they have to be able to separate the cells that aren't making protein from the cells that are making protein. And often the way you do that is something like puromycin. You kill off all the cells, right, that don't have your favorite construct inside.
yeah,
So anyways, in this assay, so the way puromycin works is it incorporates itself into the growing
yeah,
protein, and like it incorporates itself and then it blocks that process basically. And so what they're doing here is that they measure the levels of puromycin incorporation into the proteins, as a way of measuring how much protein is being
mm,
made.
yeah,
Yeah, and normally you would do that with
yeah.
radioactivity, but we've moved
Yeah,
on.
we've moved on. We no longer accept cancer as a side effect of doing science.
Yeah, exactly.
Yeah, so
Uh,
they yeah
so yeah, here's that data, right? So it's quite a messy looking piece of data
yeah
because they're looking at all the proteins, all the little nascent protein strands are being run out on this gel, right? Where the cells have either been infected with GFP or this
yeah
NSP1. And the bar graph maybe shows it a bit clearer. You get less dark
yeah
bands in general when
yeah. I
NSP1 is
guess if you
around.
blur your eyes slightly, you can sort of see that side of that blot is darker. But
Yeah,
yeah,
I don't have to blur my eyes. I can see it. Look at that in the one, two, three, fourth figure. That huge smear, right? I mean,
oh
if they're counting that not as just a bad experimental evidence, but like that's dark, right?
yeah,
That's dark
that's
there.
dark. It makes sense. I mean they present other evidence as well to support this. So
Yes. Yeah, this is only one
yeah.
line, right? This is only
So
one.
then they look in like SARS-CoV-2 in lung cancer cells, and they see something. Again, and this is a bit more
Yeah.
clearer.
See, and we're seeing patterns too in the way that they're approaching their
Mm,
stories, right? Like they want to show us, oh, when we overexpress the cell in a system, but they also want to show when an infection happens in the
yeah,
system, right? It's still all cell culture. They kind of use maybe a more realistic cell in this case, Calu 3, right, as actual human lung epithelial or
yeah,
whatever. Um, but yeah, this is a similar approach to every story that they're going to go after, right? Is like show us what it happens biochemically, show us with the expression in a cell, show us with an infection.
yeah, uh,
And here again, they do it again. This time, Vero
yeah.
cells.
Vero cells. So trying different cells just to make sure, because I think as we've seen before,
Mhm.
some cells do change the way they express things.
Mhm.
But
And actually, here's an important control that we haven't seen. They presumably do it in all the other
mm,
ones as well, but they do this red gel, right? That's
Kumasi
oh
stain
my God, I can't remember the.
is no.
No, it's not Kumasi. It has another funny name, but it just stains proteins
Yeah,
like that are in general. It's the same as Kumasi. It's the
yeah,
same as Kumasi.
yeah. I've forgotten, but I think I know that one. But yeah, because I do remember working with bright pink gels, but I can't remember
Yeah,
for the life of
I
me. But yeah,
oh Ponzo.
Ponzo, yeah, yeah.
Ponzo.
Okay, I
Yeah.
remember Ponzo staining. Yeah, I remember that.
Yeah. But this is again, we were talking about controls as trying to like hit down, right? Like common things you can say when you just see one piece of information. What could you say about that, right? You could say, oh, well, what you did in the mock infected one is you just loaded more
Yeah,
protein, and that's why we're seeing a darker band,
yeah,
right? Well, actually, no, that's not true. They show you the Ponceau stain, which stains all protein, and that one's coming up approximately equal. So these two gels, these four lanes, they all have the same amount of protein in them. It's just that the mock infected or the SARS-CoV-2 infected, they don't have as much staining from that pyrimycin antibody, and so they have less nascently translated
yeah.
proteins.
And then they
Okay.
get to the RC. So they make a mutant of the NSP1 to make it not bind to the areas where it should bind.
Mm-hmm.
So,
Mm-hmm.
so I guess RC relates to, yeah. I mean so it relates to,
I don't know why they chose that. I couldn't figure out like what does RC stand for? Because it stands for these two mutations, K164A and H165A. They both go to alanine. So that's a really common strategy when people do
mm,
mutagenesis is that like amino acids that have some sort of like charge property, positive or negative, they change them to a neutral
yeah,
one. Because as we were speaking about before, things that are sticky, right? Like they usually have some sort of charge. They might have charge
yeah,
to them, or charge is one of the ways things can stick together. If you make them alanine, then maybe they won't stick anymore.
yeah. And so the question is, what will this do? Will this stop it binding? And apparently it does. So
Yeah, so they redo, this to me, seeing them redo their binding experiments over and over again with different things, like this lab is like they're set up to do this type
yeah,
of RNA high throughput sequencing. They have the methodology and the sort of like know-how to keep going after this. Yeah, so they do it
yeah. I think
here.
that's an important thing because usually when we see these things, it's like takes 4 years to like make a paper from because the PhD student goes through to the end of their and then they publish. Whereas this has been what maybe like just under a year, like
Yeah,
6 months or
probably under a year, and they've have created like a huge like story about this from beginning to
yeah,
end. Sort of. Like mostly it's still the beginning, I would say, but it's like from beginning to like strong suspicions, ready to dive in deeper. Another control that they put in here is Delta 124 and 125. That, again, someone could say, well, maybe you just removed charged amino acids, right?
yeah,
Like the overall charge of the protein was changed, right? But they're arguing it's the specific locations of these that are important. So here they just remove two amino acids that are charged from another part of the protein, and they still see that those ones work cross-link the same way to 18S RNA, but it's only when you remove those two charged in the very specific positions do you see a difference in the way that it binds to 18S.
yeah,
Okay, so they're gonna throw in a reporter.
yep,
They're basically gonna take their mutant now through the same similar
yeah.
tests
So
as they did
does
before.
does he? So now we're going to look at all of splicing. Does it change the splicing now? And we're seeing that no, it.
No, no, I think this is still
Sorry, yeah, this is
expression.
expression. Sorry, I got confused with the other paper that we had before. This is protein
Yes,
expression.
yes. Yeah, it's just that they're taking their mutant through the
Yeah,
paces, right? They want to make sure that like yeah, that their hypothesis is the specific binding to this region that's causing the inhibition of translation. So like
yeah,
show us the money.
and
And yeah, it looks
yeah,
good. And similarly, that other control mutant that they make, the 124 and the 125, yeah, that one is still doing its thing.
yeah. So I think they've made a reasonable argument that yeah, it's this binding to this part of the ribosome. But
Hmm.
this is where like
Hmm. Hmm.
scientists who are working like, oh well, how what practical use does this have? Well, it's
Well, let's bring out our friend
ISG.
ISG
Yeah,
reporter cell again. And once again, it's the same idea, right, as the other
yeah,
one. If you break up the stuff, you could break up meaningful signaling networks, right, that help repel
yeah.
viruses.
No, wow.
So in some ways, it's like a chaos thing, right? It's like a scorched
Yeah,
earth like type of a strategy here that the virus is using.
yeah. No, I mean before this, I didn't think any of this was important, but now I see it affects interferon specifically. Oh wow, I am on on it now. It's-
And they also do some other stimulations.
yeah,
So I mentioned before the reporter ISG, like that's like a well-known
Mm.
reporter. It's kind of like artificially made, you could say. I
Yeah.
mean, everything's super artificial when it's in cell culture, but there's another system. You can also look at A549 cells, which are a type of epithelial
Yeah.
lung cell, and you can treat them with poly-IC, which is sort of a pretend double-stranded
Yeah.
RNA.
And
Yeah.
that should trigger interferon because there's things like RIG, which is an RNA binding protein that spots dsRNA and goes,
Mhm.
wait, we don't produce dsRNA in eukaryotes in our cells. Only
Yeah.
viruses do that.
Time to
Therefore,
react.
interferon, go crazy, be inflammatory, get
Yeah.
expressed in all sorts of different places. And
Yeah, and so also in this one, instead of looking at the readout, I think ISG there's like a fluorescence readout or something. It's something easy to
yeah,
measure. Here they're actually looking at more difficult things to measure, which is like how much interferon are these cells making, or how much is this ISG54
yeah,
upregulated, right? And so like this is like they're getting closer again to like try to say like there's real biological relevance here, right? Like we're looking at the molecules you would expect to fight off the virus, and they're being inhibited, right? When we stimulate, and then when we add in this NSP1.
yeah. And SF,
Okay. Oh, same. Oh, now they run their mutant through
yep,
it again. The same assay that we just talked about. The mutant isn't able to
yeah.
suppress the response. So these are also the same system, right? Still stimulated by poly-IC, and then they even do one where no, that's
So
something
they
else.
measured interferon B through ELISA, and then they have like
Hmm.
yeah, and measuring interferon B mRNA with like plus or minus poly IC. So
Yeah.
um yeah, okay,
Okay, so this brings us to the end of number two, right? The story that they're coming up with
yeah.
here. Uh, oh, or does it?
So
Sorry, I just
actually this
jumped
is like
really
almost like a
far.
cliffhanger because we've seen this point at this point like that it down like it's all like protein, but then again the virus needs to make protein in the cell. So how does the virus actually make protein? And that's going to be the
Hmm.
question that we're going to try and figure out because we know that there's all this disruption. The question is like how does I mean this is working against the virus. Surely the virus won't be able to produce anything. But that's what the next
Hmm.
question that the researchers are going to be looking at. So
Yeah, so S5A is their guess that the mRNAs from the virus probably have some structure ahead of them,
yeah,
right? And that structure is what's preventing them from being
yeah,
inhibited.
these thing called stem loops, which are like short parts of the mRNA that are formed double helixes, and then like they form
Hmm.
straight bits. So actually, like it makes it harder for some things to read, but or not. So it's kind of an interesting
Yeah,
structure.
this is also not this is a really logical sort of hypothesis
Hmm.
to make because lots of viruses have structures in this
Yeah,
region because lots of viruses do start shit in the cells, right? And like do like mess it up in such a way where there has to be like another mechanism to make those
yeah,
proteins.
uh,
So they have a reporter that makes M cherry,
yep,
and which is red, and then they have versions that have the leader or don't have the
yes.
leader.
And they find that the versions that don't have the leader don't get produced, but if they do have the leader sequence, so these are I mean these
Mhm.
aren't virus cells. They literally take mCherry, which is this gene, put the virus leader on it to see whether they get expressed when there's NSP1
Yep.
there, and it turns out that they do.
Yeah, they're querying the function of the viral
Yeah.
leader.
So it's like the virus has this cheat code that gets around its own way of messing around with the cell. So what is it doing? How is this
Right.
working? That's what we need to think about.
And actually they go really in depth. They actually there's three of these stem loops, and so they actually test whether or not like which of the stem loops are
Yeah. Do they
responsible.
need each of the stem loop, or do they? So what's going to be the
Yeah.
important part? So
So they just it's stem loop
yeah,
one. When they have stem loop one in it, that's what
yeah.
causes this
They only
effect.
need one, and they I think they find something. No, sorry.
Well, in the next one, they really want to know the
Yeah,
mechanism here, right? They're kind of this is a very well developed, I think, story about the stem loops because now they're like, okay, so we know stem loop one is important, but is it the distance between right where the ribosome would bind,
yeah.
right, as versus stem loop, or is it the stem loop just what it is.
Because
Yeah.
because there's the idea of like, oh, can it just bring the ribosome in on its own? So then maybe the leader sequence doesn't matter because the assembly is like hitting it and this, or if it's like binding
Yep.
something at the same time. So if it's binding something at the same time, then
Yes.
you need the protein. So NSP1, if that's bound to the ribosome, you need to have the stem loop at the right distance. Otherwise, it's not going to hit that protein. So if it's too close, it's going to be at the wrong
Yeah.
angle. If it's too far away, it's going to be like dangling off a bit, and the NSP1's
Yeah.
just going to like stop the. So it needs to be exact right
Yeah.
point.
Yeah. So it's like, yeah. So the question, yeah, how does it actually inhibit the NSP1? Does it hit the NSP1 off, or does it just go around the NSP1 and maybe bring the ribosome to
Yeah.
it? Right. Those would be maybe the two models that they're trying to differentiate here. And so they make these new constructs, and looks like it has to be
Yes.
close, right? If they introduce that little gap, it doesn't work.
Yeah, if they introduce like an extra gap, if it's at the wrong length, it needs to be that exact length in order for it to work.
Mm-hmm. Mm-hmm. Yep.
So
And that gives them this new hypothesis from figure 5F,
yes,
right? Where there's a interaction between NSP1, right, and the stem loop that prevents it from hitting the
yeah,
ribosome, and so then allows the ribosome to continue.
yeah. I mean, the ingenious thing about this is that host cells can't develop anything like this because the moment they create that double-stranded RNA, they suddenly opened up a
Oh, it would, yeah, it would like, it's not part of, yeah, there's not a lot of stem. Well, yeah, it's not true, right? Because there must be something, because aren't there stem loop structures that exist inside of like
oh
some
yeah,
of
you're
these
right.
RNA?
It's in like tRNAs and
Yeah,
stuff like that. So yeah,
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 yeah, yeah,
you're
yeah, yeah. There's something else. I know the certainly this is viruses evolve really
yeah,
fast, right? Like this is through viral evolution. We're not going to evolve something,
yeah.
right, to circumvent this, right? It doesn't exist in our bodies, so it's not like we're going to get it through the course of infection.
Yeah, so this is like a really interesting hypothesis. Um,
Yeah,
yeah,
and again, not too crazy because I think things like this are known from other
yeah,
viruses, right? Like there's a really common thing in biology we have like internal ribosome entry sites,
yeah,
IRES, and like that's like places where ribosomes can bind onto so they can make proteins. It kind of bypasses the need for some other complicated machinery that's used, and that comes from a
yeah,
virus. Like I think it's like some virus has those things. So it's like, viruses have these ways of manipulating translation because that's where they
yeah,
live. They live in the system of translation, basically, in our cells.
yeah,
Okay, are we ready for story number
yeah. Story
three?
number three: The Return of the Jedi.
Again, they're all the Empire
That all
Strikes
the Empire
Back.
Strikes Back. Sorry, yeah,
Right, there are no Jedi here, unless the virus
yeah.
is the Jedi. Then maybe
There
they're
are no
all the
Jedi,
Return of the
and we're
Jedi.
all going to end up with an unsatisfying cliffhanger at the end of it. And uh,
Okay, so in this one, they're focusing in. So again, like I want to just take this moment to say there's three stories that come from this paper, three red patches on this graph that they
yes,
pointed to, but you can see there's a lot more than three
oh
red
yeah,
patches here.
there is so
And
much.
so some of that maybe is spurulous for different reasons, right? We don't know, right? Without diving into it, you have no idea. But what we could say is that there's a lot of
Yeah,
interaction between the virus and our
yeah. I
RNAs.
mean, it's weird because you can get non-specific interactions which would cause really big like those big leaps, and then while those might not
Mhm.
be as easy to delve into, they might be important. But then you're gonna get a lot of noise looking into those. Whereas these researchers
Right.
have literally looked at some of the they picked out the important looking proteins and teasing apart the
Mhm.
interactions. But there is a lot that can be drawn out of this data set.
Yeah. And this isn't even the highest enrichment
No.
ones, right? Like if you look at NSP12, those are all like black all the way through these huge genes,
Yeah.
right? So like what's NSP12 doing with June, right? So immune regulation and HSP's protein folding, right?
Yeah.
Like what there's definitely like a story there as well. Maybe it's already known to some degree because maybe NSP12 is better
Yeah.
studied. But anyways, I just want to say there's lots of stories that could come from this. And this paper is like already three stories, right? They chose the ones that I guess they were able to do, maybe were most interesting to them.
Yeah, and this is they're looking at the signal particle complex, I think, or signal.
Hmm.
So
That's right. Yeah. So 7SL is from this thing called the SRP.
Yeah.
Another machine basically in our cells that is a combination of proteins and
So
RNA.
yeah, it recognizes. So proteins need to go to different parts of the cell, and sometimes they have little signals that tell the ribosome where it goes to cell. But the ribosome itself can't do that. It needs another signaling part, uh,
Yep.
signaling like kind of molecular machine that sticks on the top of it. And when it sees like this little signal, it kind of drags the ribosome over way to the endoplasmic reticulum, and then so that forces
Yep.
it to go into the membrane, and through that it can be
Yep.
translated to be shown outside the
Yeah.
cell. So this is kind of
Yeah, so this is very important for proteins that make it to the outside, either as secreted or embedded in a membrane.
yeah, exactly. So yeah, this is going to be very important, especially if you're interested in interferon, which is an expressed protein. So
Absolutely, absolutely. Good
yeah,
foreshadowing. Although I guess they foreshadowed in every
yeah,
right. We've already seen it in the in the two prequels to this,
we,
so we know that we're going down
we
that
know
path.
that we were going to be going with interferon, but
Yeah.
yeah,
Yeah, so here again, those graphs that we've seen many times, right, showing that frequency, right, of the stop region is increased in the 7SL for these two proteins, NSP8 and NSP9. And then they show us in a nice 3D model where we would normally see SRP interacting with
yeah,
these. And it's two areas, one which prevents it. Basically, they prevent the whole particle from coming together
yeah,
properly.
yeah. That's basically it. So yeah,
Yeah.
yeah, that's and so going on to the next.
But they go into more detail. They also find that NSP8 binds another region,
Yeah,
the 28S
which is
rRNA, so a part of the
yeah,
ribosome.
which kind of like because these proteins would be in almost like in a conformation with the ribosome and the signal recognition complex, I think it's called. Um,
Absolutely. Yeah. Go to the next figure. I think there's a good picture of that.
yeah,
So I mean, it's complicated, right? But I think all the pieces are here, right? 18. Sure, they have the
yep,
28S of 28S. That's the region that they found, right, is binding up to NSP8,
yeah,
right? And that region is part of the ribosome, but it also comes into contact with the 7SL,
yeah,
right, of the SRP.
yeah.
Yeah.
So
Yeah. So it's the blue, it's the gray, right? It's the gray line in the gray mRNA line, right? That's coming through, or not mRNA, the gray ES27 line, right? That goes all the way around, and you can see there the brown 7SL, right, comes into contact there.
yeah,
So like this is all happening during co-translationally
mm,
during that translation event, right?
right,
They all have to come together in order to coordinate because you wouldn't want a protein that's meant for the membrane to just get spat out into the
yeah.
cell
It
cytoplasm.
wouldn't do any, it
Yeah.
wouldn't help anyone if you want to send out a molecular signal, you want it to go into down that certain direction. So you need to have this.
Yeah, the right place.
Yeah, uh, yeah, and this.
So it's all it's like so there's how many there's the mRNA that we're gonna make, right?
Yep.
There's the ribosome, there's the single recognition particle, right?
Yeah.
And then there's the nascent peptide coming out. All of those things have to be coordinated to get proper protein trafficking.
Yeah.
I
And
guess is the thing.
and SARS-CoV-2 messes all that up with NSP8 and NSP9 because they go in.
Yeah, at least yeah, their hypothesis, right? Because they bind these areas that are basically at the juncture of these of all the things that are coming together.
Yeah. Yeah. And then they show that in microscopy. So again, they.
Well, they're going to say localization. Where does it go?
Yep. And they see that it's kind of like in and around the nucleus a bit, but it's not in the nucleus. Sorry, it's kind of surrounding it. It's-
Mm-hmm.
Um,
All over the place, actually. I mean, I see it in the nucleus too,
yeah, I think it is in. So yeah, it just seems to be all over the place. Um,
all over the place. But more important for their hypothesis in the
yeah,
cytoplasm, right?
in the
Like,
cytoplasm, which is where you'd kind of expect that to be, because it.
yeah,
Um,
yeah. Sorry, NSP9 I feel like is all over the place. NSP8, I agree, it's excluded from the nucleus.
yeah, Yeah, definitely.
And they do it again in Vero cells.
Yep. Again, we're using their antibody, which they kind of show a bit better. And yeah, this one kind of shows again that it's out on the cytoplasm a lot. Uh,
Hmm. Yeah, this one's a lot more clear.
yeah,
Um, this is probably because this is the infected
yeah,
version, right? So they're not making like in overexpression, right? When you're just making it in a cell, you're making way
yeah,
more than would normally exist. So sometimes you do find that protein in places it might not normally be. That's another nice reason to do these infection experiments because we're sort of seeing maybe more infection relevant levels of protein where it goes.
yeah,
Okay, so here's their hypothesis, right? Basically, what we said, it gets in there and it blocks the SRP from being able to recognize um, this region.
yeah,
And so the protein just like gets shot out wherever.
yeah. And this is very important if you're interested in interferon, or like other, actually not just interferon, but a whole raft of different secreted cytokines. Uh,
Yeah, tons of things use the SRP
yeah, I mean,
to get out.
because again, if you need to signal to other cells, then you need to get those proteins out. But if COVID is like stopping that from happening, then suddenly that process is interfered with, and so they
Yeah. Yeah. So this is like a prediction, right? They have like it's sort of known from uh gazing at sequences for so long, right? Like what proteins fall under the secreted pathway and
yeah
what
they've got
proteins
like
don't.
that signal peptide that is recognized by the signal recognition particle.
Hmm.
And so thus you can predict like the amount that these are going to be spliced out. And so
Totally.
you start seeing another reporter they're using, which is a let me see whether I could write it down, but it's
MGFR,
yeah
that one.
NGFR, which is a it's a neuron growth factor receptor, I think.
Ah, yes.
So
Hmm.
it's so
I think it's just secreted by cell
yeah,
cultures, right? It's like very well known secreted protein
yeah,
that they fuse to GFP.
yeah,
So that the idea is that, yeah, if you get it, this protein would be secreted, it gets secreted with GFP.
yeah.
And you can see the glowing. Hmm.
So the basic thing, if these are having effect, then you see the glowing disappear, and that's kind of what
Hmm.
they see. So they found with the membrane reporter that NSP1 doesn't have any effect on this, which is quite a good use of a control. And also they compare it to siRNAs to interfere with those direct parts of the signal recognition particle. So just as a comparison, so we can see
Hmm.
that they both kind of act and have a similar outcome.
Yeah. Yeah. I mean, again, this is like when they use the spliceosomal inhibitor,
Hmm.
right? And they say, oh, it has the same pattern,
Yeah,
right? It's like if you block, if this is like the positive control, if you destroy the thing that you know, right, would break the process, does it work in your assay? And it
yeah.
does.
And I guess NSP1 would be the negative control because it's showing that NSP1 doesn't do the same thing. Or so, yeah.
Exactly. Exactly. Hmm. Hmm.
Uh,
Okay, so then they use their sunset
Sunset.
technique
Yep.
adapted for membranes. I was trying to read about this because I didn't understand how, and I still don't understand. This is like an open question for me. I thought puromycin stops the chain,
Yeah, I think that there are
stops the growth.
ways to
Hmm.
like program escape
So why does
mutants
it
from
keep?
it for it, so that you
Yeah,
so
but no, but escape, no, but escape mutants, they wouldn't, they just wouldn't incorporate
right.
puromycin into the chain.
Okay.
So it just was weird to me that you get accumulation of puromycin on the surface. Anyways, I don't know. We don't have to answer it.
Yeah,
I couldn't figure it out
yeah,
from reading.
I yeah, I get that. That's something that I didn't really
Yeah.
get at all. I
Yeah, I trust, right?
yeah,
Because I picked up the paper, I put it in the Zotero, I picked up the paper where this method is used, and in that paper they talk about measuring membrane protein with this technique as
yeah,
well. So like it can be done, right? Like I have to trust that it can be done. It just like it never meshed with my understanding of puromycin, how that
yeah.
works, right? Because I was like, I thought puromycin stops things. So like, does that mean you have like half done proteins that are sticking out of the membrane, and that's what it's measuring? Or is it something else? And I'm not
Yeah,
sure.
that's an interesting like kind of thing to look into because yeah, I think it's more like a barrier of my own understanding rather than something that is.
It's
So
definitely. I mean, this is just like there's so much technique going
yeah,
on in here. Like it's a lot to be able to, yeah,
and we
to
like to
keep
give
up with.
ourselves like a week to relearn these entire techniques from scratch. So I mean, there are some times
Yeah,
where we will just come
totally.
up against something and be like, oh god, we needed just a little bit more time to get our heads around this.
Uh-huh.
So
Uh-huh.
uh,
So anyways, we'll say, right, we know what the readout is for,
yeah,
right? The readout is for seeing proteins that are being made, right? And in this case, they've adapted it so they're only looking at stuff on the membrane.
yeah,
And similarly, yeah, proteins are not being made on the membrane. There's no nascent chains there.
yep. And then they do some neat analysis where they break down the the cells into, so this is 7C and D, where they look at the membrane reporter, but break down by comparing it to levels of spike. So with more spike,
Mm-hmm.
you get less membrane reporters. So spike is going to be their proxy for how much SARS-CoV is in that cell, or how much the virus
Infected.
is
Yeah.
kind of producing proteins in that cell. And so
Yeah,
the more of
absolutely.
those virus proteins, the lower the amount of membrane reporter, which feeds into that idea
Mm-hmm.
that the virus is stopping the expression of this.
Yep.
And
Yeah, absolutely. Yeah, this one's like I think it's like the challenges here are like they can't see like the bulk levels of protein. They want to very specifically say, right, proteins that make it to the surface.
yes,
Um, so yeah, I think the mem using NSGR is like the important part. And I think they can see that stronger response with the spike levels. It kind of removes like the averaging issue,
yeah,
right? Like they can bin it by spike level and then see that more dramatic effect.
yeah. I mean, it could be that they should sort of very subtle effect at first, and they had to like bin it in order to quantify it.
Yeah, yeah.
But I mean, that's still like doesn't necessarily invalidate the result. That just shows us more detail. And
Yeah,
yeah,
absolutely. Absolutely. Yeah, so 7CD, right? That's what they're looking at
yeah.
in Vero cells.
And five, yeah,
Okay.
and yeah, 7CD. They yeah, again they doing the same sort of thing, and then you're getting a lot more detail as well because they did
Hmm.
flow cytometry to capture spike levels as well as so the ratio is NGFR to GFP. So GFP is like their
Hmm.
baseline, and the lower amount of NGFR, the bigger the spike on the earlier the spike will appear on that graph.
Hmm. Hmm.
I'm sorry, I'm using spike a bit more. So I mean peak on the graph, and
Yeah, spike for the spiking in the density. Yeah,
yeah,
I got
yeah,
what
yeah.
you mean.
The way they formatted the figure kind of because literally at the peak they put spike on top of it. I go, and I'm immediately going, oh wait, that's what I use. That's the thing I would say. You say that. So it's not my fault. They tricked me.
Yeah, and also in Calu 3 cells, they see the same
Yeah,
thing.
they see the same thing again, confirming it in multiple different cells, which is important.
Hmm.
Um, and then that brings us to figure 7E, which also indicates, so that kind of hints that there might be an issue with, because a lot of the SARS-CoV proteins also have this signal recognition peptide. So the
Hmm.
question is, so like spike protein is probably the most important one. So the question is, where does that get trafficked to? Um,
Yeah. They don't answer that question though, I don't think.
they don't, and I feel
I think they just throw it out
yeah they
there.
throw it out there.
That there are these three proteins, S protein, ORF7A, and ORF8. Those all could be impacted by this. I guess we know that spike isn't impacted by it, right? Because of the correlation that they're finding. More
Yeah,
spike, right, has more of this difficulty in getting membrane proteins out. So the mystery is still open, right? How does spike make it to the surface?
yeah. I mean, I feel like this is where that fancy replication organelle comes in because then it
Oh, it doesn't have to go. Yeah, it doesn't have to go into the ER
yeah.
version. It goes through this other membrane.
Yeah,
Yeah.
so there
Hmm.
is that where because I mean we've seen in previous papers where SARS-CoV-2 creates its own little miniature organelles inside the cell where it does its replication. So it could easily be, and of course where the
Absolutely.
virus is formulated,
Yeah.
that's not virus isn't formulated in the endoplasmic reticulum. It's formulated in liposomes. So actually, the trafficking
Yeah.
might not be that much of an issue for things like spike protein because they're going through a different route.
Yeah. I mean, a really nice thing would be to see where does NSP7 and 8 localize,
Yeah,
right? In response to those replication organelles,
yeah,
because maybe they're not inside of those. And then that would be a nice support
because I
for the
think like
hypothesis just provided.
in those like in those like images, Microsoft images, so those the NSP like kind of expressions seem very punctate. They seem to be like
They
in
are very
yeah,
punctate.
very
Yeah.
very small patches of it. So that could be indicating that they're in some kind of vesicles or liposomes, in which case, but
But you wouldn't want them to
no,
be
that's
there, right?
right.
You'd
You're
want them to be in where the host is doing stuff,
yeah,
not where the virus is.
yeah.
Yeah.
So that
But I mean, I know that there is some level of cytoplasmic organization in host cells as
mm,
well, right? So like the punctate could be, yes, these are punctate regions where host is happening.
yeah.
We just don't see the other, we have no idea what the other spots in the dark
Yeah, I
are.
mean, it'd be interesting to see like the co-localization of like S protein and or like S protein production and where the NSPs are acting. See whether there's like some segregation happening there, or because if there is, then that could be a thing for the replication organelle. Or if they're in the same area, then there could be some molecular thing going on that like kind of
Absolutely.
so
Absolutely.
yeah,
And again, like, we talked about this, like, this is three stories, but there's a whole bunch of stuff still, right?
yeah,
Like, both to extend the stories that we're talking about, but also to follow up on stories they didn't even mention coming from their data set.
yeah, completely.
Okay, but like they're gonna wrap this up in a very similar way as we've seen their other stories.
Yep.
They're gonna say, "Show us
It,
the effect with
yeah.
ISG reporting."
Shows the effect. And there is an effect. It's not as dramatic as the other effects, but then with because
Mhm.
they're asking a much more subtle question with membranes. Because again, they're
Yes. Yeah. Well, the inhibition of NSP 8 and 9 is like a little bit. It's less,
yeah,
right? And actually, we saw earlier in the controls, right? They kind of use NSP 8 and 9 sometimes to say like, but NSP 8 and 9 don't affect translation and they don't affect
yeah,
splicing, right? It does affect something else,
yeah,
and that ends up contributing to this core, I guess, more complex phenotype of inhibiting ISG reporting.
yeah. And then they put it together in their model where they
Yep.
kind of yeah they just show how it can affect trafficking. How like on one
Yeah.
side
That
how it's normal, and on the other side what happens when NSP 8 9 interfere.
Yeah, so normally you have those four components coming together during translation to make sure proteins get to the right spot. But again, just the binding of very specific regions, right, where all those things come together, displaces the SRP, and those proteins get degraded.
Yeah.
Yeah, so actually, I just realized I was probably misspeaking. I was talking about this NSGR reporter, right? It's used as a ratio of GFP
Yes.
reporting, right? So like they have both GFP being made in the cell, right, and NGFR,
Yes.
right? And they have to measure both to find out like what's going
Yeah,
on.
because they want to make sure that they're not seeing like just a decrease in protein overall. They want to make sure that the
Yep.
cell's still making protein as normal, but they're not. It's not secreting it. So
Yes.
that's kind of how they're doing their comparisons.
Yeah.
Uh,
And that ends the three stories that they want to
yeah,
tell. Kind of, I mean, to me, really awesome because throughout the paper they talk about how it's known in some beta coronaviruses that these things can be changed,
yeah,
right? And from my experience knowing viruses, I know other viruses do this type of stuff, right? Like some viruses do crazy things
yeah,
and they like cut off pieces of host RNA and they stitch them to their own
yeah,
RNA, right? Like there's all these different things that viruses do because ultimately like this is the hijack, right? And there's so many evolutionary ways to go and hijack the system. And so to be able to see, right, some great examples coming from a technique like this, and even have way more things to follow up on, just gives us a better understanding of, well, druggable targets.
yeah,
I mean, if we're talking about therapeutics, we're definitely going down that path. But also understanding cell biology,
yeah,
right? Because we're breaking these systems in very specific ways. It can help us learn things about stuff we didn't even know about the systems in the first place.
yeah,
Um, like I think a good example of that in the paper is they were talking about what it's the ES27,
yeah,
right? They were talking about NSP8 binds, right, or NSP8, yeah, NSP8 binds 28S RNA, and that is in range of this ES27
mm,
region where doesn't have like the structure of the ribosome is known, but that particular part of the ribosome is super
yeah.
flexible,
It varies a
and
lot.
they've never actually, yeah, they actually really don't know where it is. And so it's kind of cool to have sort of very specific ways to disrupt that interaction, right? Because there's not a lot known about what that region is involved in,
And
right? And so like by
yeah, and it's interesting because like only a year ago, this was basically in bats or pangolins or whatever host cell, and these viruses had never seen a human cell, and then now the
Mhm.
systems have evolved are already available to hitch this E27 segment, which is very much a human specific kind of segment that has been well characterized. So again,
Mhm.
that's kind of a very interesting idea that
Yeah.
that it. I mean, evolutionary
Well, I
speak.
mean, our this is gonna sound crazy or weird, but like in some ways our lives are like the petri dish for viral evolution,
Oh,
right? And so like for them to gain a foothold, their evolution has to find some sort of elegant solution, right, to living in our system, right? And like we're not masters of our own system, right? Like we don't like we can't iterate over like millions and billions of iterations the way a virus can in our
yeah.
bodies, right? So like they're this is kind of like an experiment on human beings, and like we can learn a lot from that, right? Like from this we can say like how does that work exactly, and then how does that tell us how this other segment works, and now we're learning about ourselves when we were just studying viruses.
Yeah, I mean that's because viruses are like one of the most like I guess one of the most I guess basal life forms out there. They
Hmm.
and
Yeah.
seeing how they act on some because a lot of the processes they're acting on here are very evolutionary old processes, things that are conserved throughout various. So
Right.
it's and also like I mean thinking in terms of like a bioprospecting, like understanding how the human like kind of genome and the cell functions can be changed can be
Yeah.
interesting. Like the way that viruses manipulate and change these sorts of things um, can give interesting insights on how these molecular machines work and make it guess inform us on how we can change.
Absolutely. And
Yeah,
you were speaking, you said bioprospecting. That's really similar to like when they showed that mechanism of the stem loops to stop NSP from binding up the
yeah,
ribosome, right? To me, that screams, oh, maybe that, I mean, I don't know, but like you could imagine that that could be some sort of research tool in the
yeah,
future where it's like if you can't form the specific structure, then you don't end up inhibiting this process, right? That's like, oh, maybe those are tools that we could take out of context and put them in different systems,
yeah,
right? Like there could be a reporter construct that uses a combination of NSP1 like protein and a stem loop, right,
yeah.
to get something
I mean,
done.
what the virus is doing here is essentially it's turning your cells into an expression cell line by downregulating every other protein and then biasing the expression
Oh yeah.
of viruses. So, I mean,
Absolutely. And that's a challenge in biotech where people want to make expression cell
yeah,
lines, right? Make things more efficient. Maybe some of the answers are inside of viral biology to say, like, oh, viruses do that all the
yeah.
time to
I
us.
mean,
Let's
right
hijack
here,
some of
yeah.
our industrial
It's
lines.
just like you have to exchange out a few proteins from SARS-CoV-2 to I don't know insulin, and suddenly you can turn uh, a bunch of cells into expression cells.
Yeah. Yeah, I think to me the most famous thing is that CMV
Yeah,
promoter, cytomegalovirus, right? It's in the name,
yeah.
cytomegalovirus, right? But you'll see when you look at biotech papers, CMV pops up everywhere because it's such a great promoter that they found in a virus. Because hey, viruses want to make a lot of themselves, so their promoters, they're great. They're like always on.
Yeah, I mean, you get a lot of these great things coming out of. I mean, like, I mean, I think we're going to see a lot more of these sorts of things coming out in the future as our understanding improves. But
Hmm.
I mean,
Hmm.
just and most of the vaccines are. I mean, I think the top three vaccine candidates are derived from viruses, and
Oh
those
yeah,
viruses
yeah.
were gene therapy candidates. So
Hmm. Hmm.
it's interesting to look into how these viruses work, and then we can figure. I mean, personally, as a microbiologist, I'm just interested just for the sake of knowing it. Is
Absolutely.
but there is always an aspect of like, can I make money out of this? Finally, is there something that I can finally like pay off my student debt with? Like, kind of, yeah,
Yeah. So, anyways, I thought it was a great paper. I love like that they sort of like they're constantly from the zoomed out to the zoomed
yeah,
in, right? And they zoom out, but they tell us the story, and they zoom back out again. And they tell us the story as far as it can go. They never get into animals, right? So we never really know like in the whole infection process how this plays out, but they show us the snapshots inside of cells, right? They actually infect the cells,
Yeah.
right? So we know the process is like happening in an infection
Yeah. This is
scenario.
very much like a very biochemistry like process focused paper. So
Hmm.
actually like putting that out to that macro level might not be like as like it might not show as much because again this sort of thing would get lost to the noise because there's so many other things happening with
Hmm.
the immune system in that at that stage because you've got cell death, you've got NK cells where they'll react if there isn't enough protein expressed on cells.
Hmm.
So I mean, this might have an immune action, but it might just be for the virus making sure it makes enough copies of itself. So
Hmm. Hmm.
so
Yeah, that's true. Yeah, like the fact that they're linking it to the immune system, like that's still a very tenuous
yeah,
link,
I
right? Is what you're
yeah, that's
saying,
what I'm saying. I'm
right?
saying
Like,
that basically like that could just easily be a side effect of the fact the virus is just balking the entire immune system to power its own creation, which is something that all viruses kind of want to do, and linking it to
yes,
the immune system,
yes.
I feel like that's the kind of thing that if I was a reviewer who really wanted to just mess with people, that's what I'd ask them to do on the paper. Just be like,
Yeah,
yeah,
yeah, we don't know how much they put ISG in their original plan
yeah,
or how much that was like, oh, please add this so that we can like, right, like get this paper to higher biomedical relevance.
yeah. But I mean, it's an interesting side of the paper, and it's good to have just one example of a protein, just so we have that story going on. And then,
Yeah, yeah. I mean, for me, they definitely when they did the A549 cells and they did poly-IC and interferon stimulation of them to show like, I think that that is a good example of like where they fleshed it out a little bit
yeah,
more because they like tried some different stimulations. Like it wasn't just like this reporter cell, this one isolated
yeah,
incidents, right? They try to add some more, but of course, like yeah, we kind of want to see reconstituted whole immune systems before we say anything about the actual effects on the immune
yeah.
system.
And I feel like if you do some knockouts on this virus, I mean, one of the things they could do is just do knockouts on the virus. I say that's going to be quite a difficult thing because SARS-CoV-2 is very hard to deal with and handling. But
Mm.
again, seeing
Hmm.
like how that will affect on a broader scale, like, and we'll,
Yeah, put it through the mouse model, right? Like remove NSP9 or 8 or like, your favorite NSP. No, maybe the Delta
I
RC
mean,
mutant. That was a pretty
yeah,
minimal thing.
but again, you've got that thing where there's going to be a lot of noise there that you might get a lot more information out of cell culture because again, you're going to see maybe there's going to be less infections. That could be because the immune system reacting to it. There's a lot less virus coming out maybe, so you're going to have to like go
Yep.
back to the cell line and then build up from there. So
Oh, yes, for sure, for sure. Yeah, so this information will even be in dialogue with using the animal model, right?
yeah,
I agree with
I
that.
feel like the animal model might be a bit further down the line for when we understand what's going on here. Because if it's something like it produces less virus, then there might not be
Hmm.
that necessity to use an animal model because then we kind of get the whole picture without because you kind of get the idea that if it's producing less virus, then it's not going to be as infectious. You don't need to kill an
Sure,
animal to figure that
sure.
out, and I'd rather not.
Absolutely. Absolutely.
So
Absolutely.
I mean, if you
But
only use animals if you
but
need
to
to,
know but to know whether or not the contribution to the infection is from the level, the amount of virus coming out versus the ability to stamp down the immune response, right? Like that would be something that you would only be able to answer in an animal
yeah,
model.
but I feel like you can use other mutants to find that, and even like a
Mm,
dose
mm,
ratio and things that they've been doing that in like challenge. So they started doing challenge experiments in the UK. So they're actually
hmm.
starting to doing that dose ratio to see what kind of effective dosage viruses will have, which is
Sure.
kind of like hopefully it'll go well. Hopefully there won't be any problems, but
Yeah.
yeah,
Yeah. 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 Huh. That's really cool.
yeah. So
Yeah, anyways, that's all I want to say about this paper. Do you have
yeah,
anything
I
else you
think
want
we've covered just about everything. So I want to say thank you so much to the viewers if you've made it this far. Uh, this has been a really big paper.
to? Yeah. If you guys have suggestions about things that you want us to cover, right, or questions about the papers as you read them, you want to tweet us, right?
Yep,
Either to our personals or using the hashtag microtwjc.
yeah, yeah. So thank you so much. I hopefully we'll see you again next week.
Mm-hmm.
Yep. All right. Bye.
See ya.